On March 6, 2025, the journal Cell published a cover story package of 3 peer-reviewed papers. The lead authors were a team based at Shanghai Jiao Tong University, with collaborators at the University of Copenhagen and Lawrence Berkeley National Laboratory. The papers carried a single digital object identifier, and the headline finding inside that package was a number that, when you read it on the page, does not feel real. 7,564. That is the number of microbial species the team identified in sediment samples, pulled from the Mariana Trench between 6,000 and 10,900 meters below the surface of the western Pacific Ocean. 89.4% of those species had never been observed by any human being in the entire history of marine biology. 89.4% of the most biologically dense sediment sample of the deepest place on Earth. Never seen, never sequenced, never catalogued. Until last year. Sit with that for a second. Because that sentence should not be possible. We have been studying the ocean for 200 years. We have crewed submersibles, instrumented buoys, autonomous probes that drift along the thermocline returning data every 6 hours, and in a single expedition, conducted between August and November of 2021 with a Chinese submersible called Fendouzhe, across 33 dives, one team multiplied the cataloged biological inventory of the deepest place on Earth by a factor that the paper's senior author, Xiang Xiao, described, when he was asked, as unprecedented. The full dataset, called MIR, the Mariana Trench Environment and Ecology Research Dataset, contains 1,194 samples and 92 terabase pairs of metagenomic sequence data. The dives covered Challenger Deep, Serena Deep, the eastern wall of the trench, and the slopes leading down into both. The seafloor cores were 227 push cores. The water samples were 12 in situ filtered seawater collections, and the organisms inside those samples, almost 90% of them, are organisms that the textbooks did not contain. Stay with me until the end of this video because what the MIR team found in the deepest sediments is the part nobody is talking about yet. And why it matters is not just for marine biology. It matters for every coastal population that lives on the rim of the Pacific Basin. Because the same trench that just produced the largest taxonomic novelty in the modern record is also, according to scientists at the National Weather Service Guam office, the International Tsunami Information Center, and the operational Oceanography community, a local source for tsunamis, not a buffer, a source. And the warning window for a locally generated Mariana tsunami when one arrives is between 10 and 45 minutes. If this has you hooked the way it has me, hit subscribe to this channel and drop a comment with where you're watching from and whether the Pacific Ocean is the body of water you can see from your nearest coast. Now, let's get into it. Part 1. The dive that should not have worked. In August of 2021, a Chinese research vessel called Tansuo Yahao, which translates roughly to Exploration 1, sailed from a port on the southeast coast of Hainan and steamed east into the western Pacific. On board was a deep-sea submersible called Fendouzhe, which translates as Striver, and a research team coordinated by Shanghai Jiao Tong University. The goal of the expedition, as written into the proposal that funded the project, was deceptively simple. Sample the sediments and the water column of the Mariana Trench at depths between 6,000 meters and approximately 11,000 meters. Recover what could be recovered, freeze what needed freezing, and bring the entire haul back to the lab. The reason that goal is deceptive is that nothing about it is simple. The Mariana Trench, for those of you who have not spent a meaningful amount of time thinking about it, is the deepest known feature of the ocean floor. Its deepest point, Challenger Deep, sits at roughly 10,935 meters below the surface. That is more than 6¾ miles straight down. The pressure at that depth is approximately 1,091 atmospheres. To translate that into something tangible, 1,091 atmospheres is the pressure that would be exerted on a small surface if you placed an African elephant on a postage stamp. Every push core that Fendouzhe was asked to retrieve had to survive that pressure on the way down, and then on the way back up, the pressure had to be relieved without the sediment collapsing the integrity of the sample. A sediment core compressed and decompressed wrong, in the worst case, becomes a homogenized slurry that is no longer scientifically usable. The engineering tolerance is small, the window for a clean sample is small, and the trench is large. Between August and November of 2021, Fendouzhe completed 33 dives. I want you to hold on to that number because 33 is doing a lot of quiet work in the sentence I just spoke. The operational deep-sea community had never seen a pace like that in the trench by any nation in any prior season of submersible operation. 33 dives in a single November is an order of magnitude beyond what the global manned submersible community had previously accomplished in the Mariana Trench in any single season. Up until that expedition, the cumulative manned dive count to Challenger Deep across all human expeditions in the history of deep-sea exploration was in the low 20s. The Trieste descent in 1960, the Deep Sea Challenger dive in 2012, the Five Deeps expedition led by Victor Vescovo in 2019, the follow-on dives in 2020 and 2021 by Culloden Oceanic. Add them all up, and you get a number that is comparable to what the Fendouzhe team accomplished in 3 months. The pace was not what the operational community considered normal. It was the new pace. And here is the part nobody outside of the operational deep-sea community is saying out loud. The Fendouzhe expedition was not just trying to revisit places we had been before. The push cores were programmed to land in specific, scientifically chosen locations along a depth gradient. 6,000 meters, 7,000 meters, 8,000, 9,000, 10,000, and then, in the final sequence of dives, the deepest 200 meters of the Challenger Deep floor. The pattern of the sampling was not exploratory. It was systematic. Think about what that means. A systematic sample is a sample that lets you compare like to like. A systematic sample is a sample that lets a statistician say, with confidence, that what is in the 7,000-meter sediment is similar to or different from what is in the 10,000-meter sediment. A systematic sample, in other words, is the sample that lets you answer a question, and the question that the Mir team was trying to answer was the question that, until 2021, no expedition had ever had enough data points to attempt. Is the Hadal zone, the zone of the ocean below 6,000 meters, biologically and microbiologically distinct from the rest of the ocean? Or is it just a continuation of the abyssal sediments that we have been studying since the 1970s? The answer arrived in the lab, and the answer was not what most marine biologists had assumed. When the sediment cores came back to Shanghai Jiao Tong, the team handed them to a sequencing laboratory that processed the genetic material using metagenomic protocols. The methodology, for those of you who have not spent time inside a metagenomics workflow, is this. You take a sediment sample. You homogenize it. You extract every fragment of DNA inside that sample, from every organism. regardless of whether the organism is intact or alive or recoverable as a discrete cell. You sequence those fragments. You assemble the sequences back into genomes, computationally, using software that compares overlapping fragments. And then you ask the question, of every genome you assembled, does this genome match anything in the existing global reference database? If yes, you have identified a known organism. If no, you have identified an organism that science did not know existed until your lab opened the sample tube. The numbers that came back from the Mir metagenomic pipeline were, when they arrived in the form of a draft paper sent to the Journal Cell in late 2024, the numbers I read at the top of this video. 7,564 total microbial species identified. 89.4% of those species were unmatched to any existing global reference database. Read those two numbers again because they are doing a lot of quiet work. 89.4% of 7,564 organisms are organisms that, prior to the Fendouzhe expedition and the Mir sequencing, did not exist in the cataloged biological inventory of Earth. They existed in the trench. The trench had them. But the trench had not been sampled densely enough, or systematically enough, for any human to know they were there, until November of 2021, and then until March 6th of 2025, when the papers cleared peer review, and the Mir Consortium published its results. The cover of Cell that week showed an artist's rendering of the trench wall, with the depth gradient marked along the side, and a stylized representation of the Fendouzhe submersible descending the slope. The accompanying editorial described the Mir dataset as a foundational reference resource for the Hadal zone, comparable in scale and ambition to the Human Microbiome Project. That is not a casual comparison. The Human Microbiome Project, which was funded by the National Institutes of Health beginning in 2008, produced a reference inventory of the bacteria, archaea, and viruses living on and inside the human body. It rewrote textbook microbiology. It changed how clinical medicine thinks about chronic disease, and the comparison to Mir in the Cell editorial was not metaphorical. The editors meant that the Mir dataset is a reference resource of equivalent scientific weight. The implication, if you allow yourself to follow it, is that what we did not know about the Hadal zone before March 2025 is roughly what we did not know about the human body before 2008. Almost everything. Now, I want to be precise about something because precision is going to matter for the rest of this video. The number 7,564 is the number of microbial species. Microbial means bacteria and archaea primarily, not multicellular animals, not crustaceans, not fish, not anything you would normally think of when you imagine deep-sea life. The biological inventory that Mir is rewriting is the microscopic inventory, the single-celled life that lives in the sediment, that processes nutrients, that respires sulfur or methane or hydrogen, that converts dissolved iron between oxidized and reduced states, that decomposes the organic snow that falls from the surface ocean and lands at depth. That is the inventory that Mir found, sequenced, and published. 7,564 of those single-celled organisms. 89.4% of them new to science, and every one of them is a node in an ecological network that, until 2025, the operational marine biology community could not see. That fact alone is the headline. That fact alone is why I am spending the next 2 hours of your time on what is, on the narrow question, a paper about microbes. But the headline is the beginning of the story. It is not the story. The story is in the second-order finding. The story is in what the Mir team observed when they looked at the patterns inside the 7,564, because what they observed when they sorted the species by depth, by sediment chemistry, by ambient pressure, by trench geometry, is that the Hadal microbial community is not a continuation of the abyssal microbial community above it. It is its own thing. It has its own rules. It has its own dependencies, and the rules it follows, the dependencies it has, are coupled, mathematically and ecologically, to a geological process happening directly underneath the sediments where Mir sampled. The Pacific tectonic plate. Bending, hydrating, subducting into the mantle, and carrying the chemistry of seawater down into the Earth's deep interior at a depth where, on geological timescales, that chemistry returns to the surface as volcanic outgassing thousands of kilometers away. The Hadal zone, in other words, is not a place where life is barely surviving. It is a place where life has organized itself around a planetary-scale geological engine. The engine that builds and recycles the lithosphere. The engine that drives the slow chemistry of the deep earth. The engine that, when it slips, generates the kind of seismic events that move continents. That is the part nobody in the marine biology press is talking about. That is the part this video exists to walk you through. And to walk you through it carefully, we have to start with the data underneath the data. We have to walk through what 89.4% of 7,564 organisms actually means. That is part 2. Part 2, the 89.4%. When a marine biologist tells you that almost 90% of the organisms in a sediment sample are new to science, the first thing you should ask is, new compared to what? Because the percentage is meaningless without the comparator. And the comparator, in the case of the Mir dataset, is not the inventory of life on Earth in general. It is the inventory of life on Earth that has been sequenced, deposited in a public database, and made queryable to a metagenomic pipeline. There is a distinction worth drawing carefully here, because the distinction is the difference between an exciting paper and a paper that resets the foundations of a field. The catalogued microbial inventory of Earth, as of 2024, was housed primarily in 3 reference databases. The first is GenBank, which is maintained by the National Center for Biotechnology Information, part of the National Institutes of Health in Bethesda, Maryland. The second is the Sequence Read Archive, which is also maintained by the same center. The third is the European Nucleotide Archive, maintained by the European Molecular Biology Laboratory at the European Bioinformatics Institute in Hinxton, England. These 3 databases together hold the genetic sequences of every microorganism that any laboratory anywhere in the world has uploaded to a public repository over roughly the last 30 years. The total count of distinct microbial species represented across all 3 databases is somewhere in the range of 1.3 to 2 million, depending on how you count and how you handle redundancy. When the Mir team ran their assembled Hadal genomes against those 3 databases, the matching algorithm returned a result for every genome. 89.4% of the Mir genomes did not match anything in the global reference. Not at the species level, not at the genus level in many cases. Some of them did not match at the family level. Think about what that means. A genome that does not match anything at the family level is a genome from an organism that is so genetically distant from anything that has ever been sequenced that the algorithm cannot place it in the existing tree of life. It is, by definition, new biology. It is biology that the operational marine microbiology community had never seen, in the strict sense that no sequencing run anywhere in the world, in the 30 years that public reference databases have existed, had ever returned the data this team was now holding in its hands. Now, I want to be honest with you about something, because honesty matters when the headline number is this big. The fact that an organism is genetically distant from anything in the public reference database does not, by itself, mean the organism is exotic in some metaphysical sense. The reference database is biased. It is biased toward organisms that have been studied for medical reasons, agricultural reasons, environmental monitoring reasons. It is biased toward the surface ocean, the gut microbiome, the soil environments of the temperate latitudes, the marine sediments of the continental shelves. The Hadal zone, until Mir, was sampled so rarely that almost any sufficiently deep sample would have returned a high novelty percentage. That is not a critique of the Mir finding. It is a contextualization of it. The Mir finding is that the Hadal zone is undersampled, to the point that systematic sampling of it returns almost a complete novel inventory. The implication is not that the trench is some kind of cosmic anomaly. The implication is that the trench is a place we have been ignoring, and the cost of ignoring it is that the existing reference for the chemistry, the ecology, and the genetic diversity of the deep ocean is approximately 89.4% incomplete. That is a different sentence than the headline. But it is, I think, the more honest sentence. And it is the sentence that, when you sit with it for a moment, has all the same implications as the headline. Because if 89.4% of the cataloged biology of the planet's deepest ocean is missing from our reference databases, then every model that has been built using those reference databases has been built on an incomplete picture. Models of carbon cycling that assume the deep ocean is a passive sink. Models of the global nitrogen cycle that treat the Hadal zone as a black box. Models of how the chemistry of the surface ocean is regulated, over centuries and millennia, by the slow exchange with the deep ocean. All of those models have been built on, at best, 10.6% of the relevant biological inventory. And now, with Mir published, the missing 89.4% has begun to be inventoried. Pay attention to this because this is the part of the story that most coverage of the Mir papers in the mainstream press got wrong. The mainstream press treated the Mir discovery as a curiosity. New species in the deepest trench, what a cool number. Here is a picture of an amphipod. That is the frame. The frame the operational biogeochemistry community uses is different. The operational frame is that the global ocean biogeochemistry models have just received a foundational input, and over the next 3 to 5 years, those models are going to be rewritten to account for the new inventory. And the rewrites, when they are published, are going to change every downstream projection that depends on the deep ocean, including projections of how the ocean absorbs atmospheric carbon, including projections of how dissolved oxygen is distributed at depth, including projections of how the chemistry of the deep ocean is going to respond to surface warming over the next 50 years. That is what the Mir team published in March of 2025. They did not publish a curiosity. They published the missing 89.4% of the input data for the planet's largest biological black box. Let me walk you through the methodology a little more carefully, because the methodology is the part that lets you, the audience for this channel, evaluate the result for yourself. The Fendouzhe team did not just collect sediment. They collected sediment with a depth-encoded push core system that records the depth of every subsample within the core. A push core, in the context of the Mir expedition, was a vertical sediment column roughly 30 centimeters tall, recovered intact from the trench floor and immediately sealed against the surrounding water. When the core was returned to the surface, it was sliced in the laboratory at 1 centimeter intervals. Each 1 centimeter slice was then assigned a depth tag, corresponding to the depth below the sediment-water interface where that slice originated. The slice was then divided into chemical analysis fractions and biological analysis fractions. The biological fraction was the input to the metagenomic sequencing pipeline. What this means, in practice, is that the mere dataset is not just a list of organisms found in the trench. It is a depth-resolved inventory of organisms found at specific calibrated depths within the sediment column at specific calibrated locations on the trench floor. The dataset is structured like a three-dimensional volumetric reconstruction of the microbial life in the trench. You can ask it questions like, what bacteria are present in the top 1 centimeter of sediment at 9,000 meters depth, versus what is present at 10,000 meters depth, versus what is present at the same depth but in a different region of the trench floor. The dataset can answer those questions because the data is collected in a way that lets it, and the answers that the dataset returned when the MIR team began to query it were the answers I previewed at the end of Part 1. The Hadal microbial community is its own thing. It has its own rules. It has its own dependencies. The dependencies are coupled to a geological process that operates at a depth and a scale that, until very recently, was treated as essentially decoupled from biology. Now, I want to be precise about how the Hadal microbial community differs from the abyssal community above it. The abyssal zone, for context, is the zone of the ocean between roughly 4,000 and 6,000 meters depth. The Hadal zone is everything below 6,000 meters. The break between abyssal and Hadal is not a physical wall, it is a geographic threshold, because below 6,000 meters, the only ocean environments that exist on Earth are the trenches. The deep ocean basins, away from the trenches, almost universally have floors that lie between 4,000 and 6,000 meters. Below 6,000 meters, you are in a trench, by definition. So the Hadal zone is the zone of the trenches, and the MIR dataset is the first systematic sample of the microbial life of that zone. What the team found when they compared the Hadal community to the abyssal community was a discontinuity. Not a gradient, a break. The species that dominate the upper Hadal zone, at depths between 6,000 and 7,000 meters, are largely different from the species that dominate the abyssal sediments above. The species that dominate the deep Hadal zone, at depths approaching the trench floor itself, are different again. And the species that dominate the deepest Hadal sediments, at depths approaching 11,000 meters in Challenger Deep, are different a third time. Each layer is its own ecological assembly. Each layer has its own dominant genera, its own metabolic profile, its own carbon source, its own electron acceptor chemistry. The trench is not a single ecosystem. It is at least 3, layered vertically, with each layer hosting its own community of organisms adapted to its own specific combination of pressure, temperature, available nutrients, and incoming chemistry. Read that again because the implication is bigger than it sounds at first read. If the Hadal zone is at least 3 layered ecosystems, vertically stacked, then the carbon and nitrogen cycling at the bottom of the ocean is not a single process. It is at least 3 processes operating in series, each transforming the chemistry of the sinking organic matter before it reaches the next layer. The carbon that arrives at the trench floor, 10,000 meters down, has been processed by at least 3 distinct microbial ecologies on the way down. And the chemistry of what reaches the floor is not the chemistry of the surface ocean. It is the chemistry that emerged from that 3-stage processing chain. This matters because the chemistry that reaches the trench floor is then available to be subducted, to be drawn down with the Pacific plate as it bends into the mantle, to be carried into the deep earth and, on geological timescales, returned to the surface as volcanic outgassing along the volcanic arcs of the Mariana Islands and beyond. The chemistry of the Hadal zone is, in a sense, that no one in the geochemistry community was talking about before Mayer, the chemical input to the deep earth. And the Mir team has now, for the first time, given us a systematic inventory of what that input actually is. I want to underline this because it is one of the parts of the Mir story that the mainstream press completely missed. The deep ocean is not just a place where organic matter falls and accumulates. The deep ocean, where it meets the trench floor, is a place where organic matter is processed by a multi-stage microbial ecology, and then subducted into the planet's geological cycle. The Hadal zone is the interface. It is the surface contact between the surface biosphere and the deep earth. And until Mir, that interface was, in scientific terms, almost entirely unsampled. Now, the Mir team did not publish just the species inventory. They published an analysis of the metabolic capabilities of the Hadal community. Which organisms can respire sulfur? Which organisms can fix nitrogen? Which organisms can metabolize methane? Which organisms can transform iron between oxidized and reduced states? The metabolic profile of the Hadal community is, when you read the supplementary materials of the Mir paper, almost uniformly different from the metabolic profile of the surface ocean. The dominant respiration pathway in the Hadal sediments is sulfur-based. The dominant nitrogen fixation pathway is mediated by genera that are vanishingly rare in surface ocean samples. The dominant carbon source for the deepest sediment communities is, in the Mir team's analysis, organic carbon that has been chemically transformed by the upper Hadal layer before it reaches the bottom. The Hadal zone, in other words, is not just a place where surface organisms have adapted to high pressure. The Hadal zone is a place where a distinct ecological assembly has evolved, with its own chemistry, its own metabolism, and its own ecological rules that processes the input from above into the chemistry that gets subducted below. That is the 89.4%. That is what almost nobody is saying out loud. And that is the foundation we have to lay before we can talk about why the same trench that produced the Mir discovery is also, according to the operational tsunami community, the part of the Pacific Basin that you should be paying the most attention to. Because the Hadal microbial community is sitting directly on top of one of the most active subduction zones on the planet. And the same processes that are feeding the microbes are also, on a different timescale, generating earthquakes. The two are not unrelated. They share a single mechanism, and that mechanism is the subject of Part 3. Part 3, the hydration line. To understand why the Mir team's biological discovery is connected to seismicity in the Mariana subduction zone, you have to understand the geometry of the subduction zone itself. And the geometry of the Mariana subduction zone, for reasons that geologists have been studying for decades, is unusual. A subduction zone, for those of you who have not spent time inside a structural geology textbook, is the region of the Earth's surface where one tectonic plate slides beneath another. The Pacific Plate, which is the largest tectonic plate on Earth, is in slow motion to the northwest. As it moves, it encounters the smaller plates that form the western boundary of the Pacific Basin. In some places, the Pacific Plate slides beneath those smaller plates at a relatively gentle angle. In other places, the angle of descent is steeper. The Mariana subduction zone, where the Pacific Plate slides beneath the Philippine Sea Plate, has the steepest subduction angle of any active subduction zone on Earth. At its steepest, the descending slab is going down at angles approaching 90 degrees, almost vertical. The geometry matters because the geometry controls everything else. The depth of the trench is controlled by the geometry. The pattern of seismicity is controlled by the geometry. The volcanic arc behind the trench, the Mariana Islands, is controlled by the geometry, and the way water is carried from the surface ocean down into the mantle, which is the part this section is about, is controlled by the geometry. Here is the part the operational seismic and geochemistry communities have been quietly building consensus on over the last decade, but which has not made it into the mainstream coverage at all. The Pacific Plate, as it enters the Mariana Trench, is not dry. It is hydrated. Water from the overlying ocean has, over millions of years of slow exposure, infiltrated the plate through a network of fractures that develop as the plate bends downward into the trench. These fractures are called outer rise faults, and they are visible in the bathymetric data as long, linear depressions on the seaward side of the trench axis. Each outer rise fault is a place where seawater has been able to penetrate into the upper kilometers of the oceanic crust, hydrating the rock as it goes. A paper published in 2025 in the journal Tectonophysics, in a study titled Along Strike Variations in Intermediate Depth Seismicity in the Southernmost Mariana Subduction Zone, documented the pattern. The Pacific Plate, as it enters the Mariana Trench in its southernmost segment, is more hydrated than the same plate in the central or northern segments. The hydration shows up in the seismicity data as a specific pattern of intermediate depth earthquakes. Intermediate depth, in the language of seismology, means earthquakes occurring at depths between roughly 70 and 300 kilometers below the surface. These earthquakes are not the shallow earthquakes that you think of when you imagine a megathrust event. They are deeper. They occur at the depth where the descending slab is undergoing metamorphic transformations as it heats and pressurizes. When water is carried into the mantle along the descending slab, the water does not stay neutral. The water reacts with the minerals of the slab. The minerals dehydrate as the slab heats, releasing the water into the surrounding mantle wedge. The released water lowers the melting point of the mantle, contributing to the magma generation that ultimately feeds the volcanic arc above. And the dehydration reactions themselves, at certain depths, are seismically active. They generate small earthquakes as the slab releases its bound water in pulses. This is the part that has been building consensus quietly. The intermediate depth seismicity of the Mariana subduction zone is not just a record of plate motion. It is a record of water transport. Each intermediate depth earthquake is, in some sense, a dehydration event in the slab, and the pattern of those earthquakes, across the length of the subduction zone, is a map of how water moves from the surface ocean, through the trench, into the mantle, and ultimately back to the surface as volcanic activity. Now, here is where it connects to Mir. The microbial community in the Hadal sediments at the bottom of the trench is sitting directly on top of the input region of this water transport system. The sediments that the Fendouzhe team sampled are sediments that are about to be subducted. Geologically, the timeline is slow, on the order of millions of years for any given sediment to be carried into the mantle. but the chemistry of the sediment, including the chemistry imparted to it by the microbial community, is the chemistry that enters the subduction conveyor, and the chemistry imparted by the microbial community, as the MIR team documented in the supplementary materials of the third paper in their March 2025 package, includes specific transformations of dissolved iron, sulfur, and nitrogen that are not seen anywhere else in the ocean. The Hadal microbes are taking the surface-derived organic matter and chemically modifying it before it enters the subduction zone. The modified chemistry is then carried down into the mantle, where, millions of years later, it is released back to the surface as volcanic outgassing. Think about what this means. The microbiology of the trench is not a passive curiosity. The microbiology of the trench is a chemical preprocessor for the planet's volcanic outgassing system. The Hadal community is, in geological time, regulating the chemistry of the volatiles that the Earth's interior releases into the atmosphere through the Mariana volcanic arc. The biological scale of that effect is small at any one moment, but over the lifetime of the subduction zone, integrated across millions of years and hundreds of millions of kilograms of subducted sediment, the effect is not small. It is meaningful, and it is, until 2025, completely unmapped. I'm going to slow down here because this is the conceptual hinge of the entire story. The Mariana Trench is not a curiosity of marine biology. The Mariana Trench is a chemical interface where the biosphere meets the lithosphere. The microbes are the interface. The plate motion is the conveyor. The seismicity is the timing signal, and the volcanic arc behind the trench is the output. The mere discovery, in other words, is not just the discovery of new species. The mere discovery is the discovery of a previously uncatalogued component of the planetary geochemistry cycle. That is the part the mainstream press will not tell you. Not because anyone is hiding it, because the connection between marine microbiology and geochemistry is the kind of connection that requires you to read both the MIR papers in Cell and the tectonophysics paper on intermediate depth seismicity in the same week, with the same brain, and then sit down and ask yourself what the combined implication is. Almost nobody is doing that. The Marine Biology Press covers the MIR papers as a marine biology story. The Seismology Press covers the tectonophysics paper as a seismology story. The combined story sits in the gap between the disciplines and, as of May 2026, has not been told in any single mainstream publication that I have been able to identify. This channel, the audience watching right now, is one of the few audiences that is being told the combined story, and that, for the rest of this video, is what we are going to walk through. Let me get more specific about the seismicity pattern, because the specifics matter. The intermediate depth earthquakes in the southernmost Mariana subduction zone, according to the Tectonophysics 2025 paper, cluster in a pattern that the authors describe as along strike variations. What that means, in plain English, is that the earthquakes are not evenly distributed along the length of the subduction zone. They cluster in some places. They are sparse in others. The clusters correlate, in the paper's analysis, with the locations where the descending Pacific plate is carrying the most water into the mantle. The sparse regions correspond to where the descending plate is drier. The pattern, in other words, is a hydration map. The same paper documents a specific feature in the southernmost segment of the Mariana subduction zone. An oceanic plateau, a slightly thicker section of oceanic crust, is subducting at that location. The plateau, because it is thicker, brings more bound water into the subduction zone than the surrounding standard thickness crust does. The seismicity over that segment is therefore more intense. The authors of the tectonophysics paper interpret the elevated seismicity as direct evidence of the elevated water content of the subducting plateau. The interpretation, while debated in the specialist literature, has been published, peer-reviewed, and is now part of the formal record of how the Mariana subduction zone is understood. Read that one more time because the implication carries forward into Part 5 of this video. The Mariana subduction zone is not seismically uniform. Some segments are more active than others. The active segments correspond to the segments where the subducting plate is carrying the most water. The water is what enables, in the deep slab, the metamorphic reactions that generate intermediate depth earthquakes. The pattern is intelligible. The pattern is now in the published literature. And the pattern, when it intersects with the operational tsunami forecasting community's understanding of where, in the Mariana subduction zone, a large megathrust earthquake might be most likely to occur, becomes a piece of information that anyone living on a Pacific Basin coastline should at least be aware of. Now, I want to be honest with you about the size of the megathrust risk in the Mariana subduction zone specifically. The Mariana subduction zone is not the Cascadia subduction zone. The geometries are different. The plate boundary mechanics are different. The historical record of large earthquakes in the Mariana Arc is, compared to Cascadia, Japan, Chile, or Sumatra, sparse. The largest documented historical earthquake in the Mariana Arc was a magnitude approximately 7.7 event in 1849. The instrumented record, which began in the early 20th century, has not captured a magnitude 8 or larger event in the Mariana Arc. The arc is steep. The coupling between the plates may be weak in some segments, and the megathrust earthquake potential is, by the standards of the larger subduction zones, considered moderate to low at the magnitude 8+ scale. That is the honest, settled version of the science. The Mariana Arc is not a guaranteed megathrust generator on the scale of Cascadia, and anyone who tells you otherwise is overstating what the seismic record supports. But here is the part. The honest, settled version of the science does not let you skip. The Mariana subduction zone, even at its more modest megathrust potential, is still a local tsunami source for the inhabited islands directly above it. Guam and the Commonwealth of the Northern Mariana Islands, both United States territories, sit on the western, overriding plate side of the trench. The distance from the trench axis to the closest inhabited shorelines on those islands is less than 200 kilometers. A locally generated tsunami, originating from a moderate magnitude 7 or greater earthquake along the Mariana subduction interface, would arrive at the closest inhabited shorelines in between 10 and 45 minutes. That arrival window is the warning window. The warning window is short. And here is the second part, which is the part that almost nobody outside of the operational Pacific tsunami forecasting community is talking about. The same Pacific Basin that contains the Mariana subduction zone also contains the Tonga Arc, the Kuril Arc, the Kamchatka Arc, the Aleutian Arc, the Cascadia subduction zone, the Mexican subduction zone, the Peruvian and Chilean subduction zones. The Pacific Basin is, geologically, a system of interconnected subduction zones, each of which is capable of generating a tsunami that crosses the basin and arrives at the opposite shoreline in a number of hours that depends only on the speed of the wave and the distance traveled. In July of 2025, an earthquake of magnitude 8.8 occurred off the Kamchatka Peninsula in the Russian Far East. The tsunami it generated crossed the Pacific. Tsunami gauges in Hilo, Hawaii, recorded a wave of 4.9 feet relative to normal sea level. Tsunami gauges in Kahului, Maui, recorded 4 feet. The Oregon Department of Emergency Management issued advisories. The California coast, near the Oregon border, was placed under a tsunami warning that was later lowered to an advisory. The Pacific Basin is connected. The subduction zones are connected, and the Mariana subduction zone, whether or not it is the most likely originator of the next major Pacific tsunami, is one node in a network of subduction zones that is jointly responsible for the basin-wide tsunami risk that millions of people on the Pacific Rim are exposed to. The Mir discovery sits on top of one of those nodes. The Hadal microbiology sits on top of one of those nodes. The chemical pre-processing that the microbes are doing, that the Mir team has finally documented, is happening on top of one of those nodes. The trench is alive in multiple senses simultaneously, biologically alive in a way that is only now being inventoried, geologically alive in the way that has been studied for 100 years. And the combination of those two senses is the story this video is here to tell. Part 4 is the part of this story that the historical record actually does have a useful case study for. Because there has been one event in the last 10,000 years that combined a submarine geological process with a coastal population and produced the kind of outcome that you would, if you were honest, lose sleep over. That event was not in the Pacific. That event was in the North Atlantic, and it is the lens through which we can understand what the modern Pacific risk profile is actually pointing at. That is Part 4. And in Part 4, I'm going to ask you to think about what an apocalyptic flood looked like in the year 6200 before the Common Era, and what the survival numbers from that flood actually were. Because the survival numbers from the only event that comes close in the prehistoric record are not numbers you want to read on a quiet evening, but they are the numbers you should know. Part 4. What 6200 before the Common Era knew. This is the retention peak of the video, and the part where the audience for this channel will think the most carefully about what the modern Pacific risk profile actually means, by anchoring it to the only event in the prehistoric record that comes close, approximately 8,200 years ago, give or take a few generations, off the western coast of what is now Norway. 3 sequential submarine landslides released approximately 3,500 cubic kilometers of sediment from the continental shelf into the deep North Atlantic basin. The cumulative event is called the Storegga slide, after the Norwegian word for the Great Edge, which is what local fishermen historically called the underwater escarpment where the slides originated. The Storegga slide is the largest documented submarine landslide of the Holocene, the geological epoch in which we currently live. It is also, by every available estimate, the cause of the most catastrophic tsunami in the prehistoric human record. The numbers, when you read them carefully, are difficult to absorb in a single sitting. So, I'm going to walk you through them slowly. 3,500 cubic kilometers of sediment moved in a series of 3 slope failures. The head wall of the slide complex extended approximately 290 kilometers along the Norwegian shelf edge. The dropped material slid into water depths approaching 3,000 meters. The vertical displacement of that mass of sediment falling into the abyssal basin displaced an equivalent volume of water upward. The displaced water propagated outward from the slide as a tsunami. The wave heights, when the tsunami reached the surrounding coastlines, were recorded geologically. The way you record a tsunami wave height geologically is by finding deposits of marine sediment, including marine fossils, at elevations above the contemporary sea level. The deposits indicate the height to which water rose during the event. Excavations of Mesolithic-era coastal sites in northern and eastern Britain, in the Shetland Islands, in eastern Scotland, in the coastal sediments of the eastern Norwegian seaboard, have all recovered Storegga tsunami deposits. The deposits, when their elevations are corrected for changes in relative sea level since the time of the event, give you the run-up height of the wave at the location where it was deposited. On the Norwegian coast, near the slide complex itself, the run-up heights reached approximately 12 meters. That is 40 feet. A 40-foot wave is the height of a 4-story building. On the eastern coast of Britain, more distant from the source, the run-up heights have been documented at between 3 and 6 meters. That is between 10 and 20 feet. Across the Shetland Islands, the wave reached heights of at least 9 meters in some locations. In the southern North Sea, where the now submerged landmass of Doggerland connected what is now Britain to what is now continental Europe, the wave swept across a low-lying landscape that had no natural barriers to slow it. Doggerland. I want to pause on this name for a second because it is the name of a place that almost nobody alive remembers, but a place that, for several thousand years before the Storegga event, was home to a substantial Mesolithic population. Doggerland, in the early Holocene, was a landscape of rivers, forests, marshes, and coastal estuaries that connected Britain to mainland Europe. Mesolithic hunter-gatherers lived there. Their settlements have been documented by the dredging of artifacts from the floor of the modern North Sea. Fishing weirs, arrowheads, worked bone, the antlers of red deer with the cut marks of butchery. The artifacts indicate that Doggerland was inhabited, that it sustained a culture, that it was, by the standards of the time, populated. The Storegga tsunami swept across Doggerland. The wave, by the time it reached the shallow southern North Sea, was lower in amplitude than the 12-meter wave that struck Norway. But the topography of Doggerland was almost entirely low-lying. There were no hills to climb. There were no inland reaches more than a few dozen kilometers from the coast. The wave, when it arrived, propagated inland across a landscape that had no defenses. The estimates of the Mesolithic population of Doggerland at the time of the event range from approximately 700 to 3,000 individuals. The estimates of the survival rate are not gentle. The land surface of Doggerland, post-Storegga, was significantly reduced. The combination of the tsunami damage and the broader climatic context, which included accelerated sea level rise associated with the 8.2 kiloyear climate event, meant that within a few generations of the Storegga event, Doggerland was no longer habitable. The remaining land was flooded. The population was, by every reasonable interpretation of the archaeological record, eliminated. Some descendants may have migrated to Britain or to mainland Europe. The continuity of the Doggerland culture, however, ends in the archaeological record at approximately the time of the Storegga event. The land disappeared. The people who lived on it disappeared with it. Sit with that for a second, because that sentence is not a metaphor. The Doggerland population was eliminated. The wave that arrived on Doggerland's coastline was the largest North Atlantic submarine landslide tsunami the Mesolithic world had ever seen, and the inhabitants had never seen anything remotely like it in the cultural memory their grandparents would have carried, because no event of that scale had occurred in their living tradition. The land itself ceased to exist as inhabited geography, and the trigger was a submarine landslide, occurring hundreds of kilometers offshore, that generated a tsunami the inhabitants had no warning of and no possible way to anticipate. Now, the British mainland was struck by the same wave at lower amplitude. The Mesolithic population of what is now Britain at the time of the Storegga event has been estimated in the published archaeological literature at approximately 1,000 individuals across what is now northern Britain. The published peer-reviewed estimate in a Frontiers in Earth Science paper from 2021 that reviewed the archaeological evidence is that up to 25% of the Mesolithic population of northern Britain may have been killed by the Storegga tsunami. 25%. 1 in 4. At specific archaeological sites along the eastern Scottish coast, the localized mortality in places where the wave reached the inhabited zone has been documented at higher rates still. At a site called Howick on the Northumberland coast, the mortality within the resource-rich intertidal zone has been estimated at approximately 100%. Every inhabitant of the Howick coastal settlement, in the moment the wave arrived, was killed. Think about what that means in operational terms. A Mesolithic settlement at Howick was a long-term, multi-generational residence. The site had been occupied for at least a century before the Storegga event. The inhabitants would have been intimately familiar with the local coastline, with the tidal cycles, with the patterns of weather and fishing, and food gathering. They would have had a daily life that, from the inside, would have felt continuous, predictable, and survivable. And then, one morning, with no warning, a wave 6 meters high arrived along their entire coastline simultaneously, and the settlement ceased to exist. The bodies, if any survived undisturbed in the sediment, were buried under the deposits of the wave itself. The local culture, which had occupied that location for 100 years, was eliminated in minutes. The Storegga tsunami is the only event in the Holocene archaeological record that has been documented at this combination of magnitude and direct impact on inhabited shoreline. It is the event we turn to when we want to understand what a basin-scale submarine geological event can do to a coastal population. And it is the event that, when you read it carefully alongside the operational Pacific tsunami, forecasting communities' documentation of the modern Pacific Basin risk profile, becomes a calibration point. The calibration is uncomfortable. The Storegga event displaced approximately 3,500 cubic kilometers of sediment. The modern Pacific subduction zones, including the Mariana, do not, individually, threaten to displace volumes of that scale on the timescales relevant to modern coastal populations. The historical record of major Pacific megathrust earthquakes, including the 2004 Indian Ocean event, the 2011 Tohoku event, and the 1960 Chilean event, includes individual events whose tsunamis displaced volumes of water on the order of tens to low hundreds of cubic kilometers. The Pacific Basin does not have, in its instrumented record, a Storegga scale event, but here is the part that the honest scientific framing does not let me skip. The Storegga event itself was unprecedented in the local instrumented record of its time. The Mesolithic inhabitants of Britain and Doggerland had no historical memory of a similar event, because no similar event had occurred in living cultural memory. The Storegga event was, from the inside, a black swan. It was outside the operational risk model of the population it affected, and it killed 25% of that population in a single morning. The 2004 Indian Ocean earthquake, similarly, was outside the operational risk model of the populations it affected. The communities along the coastlines of Indonesia, Sri Lanka, India, Thailand, the Maldives, Somalia, and Tanzania, the communities that the 2004 tsunami swept across, had no local memory of an event of that scale. The Indian Ocean basin had not produced a tsunami of that magnitude in any living person's memory. And on December 26th of 2004, a magnitude 9.1 earthquake along the Sunda subduction zone generated a tsunami that killed approximately 230,000 people across more than a dozen countries. 1.7 million were displaced. The waves reached the East African coast. Think about what those two events have in common. Both were outside the local historical record at the time of their occurrence. Both were generated by submarine geological mechanisms that the operational science community had identified as theoretically possible, but had not, with confidence, predicted as imminent. Both produced casualty counts that were, in their respective historical contexts, unprecedented. And both, in retrospect, fit into a longer geological timescale of events that we now know occur regularly, even if not on the human timescale. The Pacific Basin has not produced its Storegga in living memory. That fact, by itself, is not reassuring. The Pacific Basin has produced events comparable to Storegga in the geological record. The Cascadia subduction zone, on the United States Pacific Northwest coast, produced a magnitude 9 megathrust earthquake on the evening of January 26, 1700. The earthquake generated a tsunami that crossed the Pacific. Japanese coastal records, contemporary with the event, documented a tsunami arrival in Japan with no preceding earthquake that local residents could feel. The tsunami in Japan reached the coast approximately 10 hours after the Cascadia earthquake, with wave heights of between 2 and 5 meters. Coastal Mesolithic era Native American communities along the modern Oregon and Washington coast were, by the same logic that applies to Doggerland, swept away. The historical archaeological record of those communities indicates that there is a discontinuity in the occupied record at approximately the time of the 1700 event. I want to be honest about the limits of this comparison. The Mariana subduction zone is not the Cascadia subduction zone, and it is not the Sunda subduction zone. The Mariana Arc has not, in the historical record, produced a megathrust earthquake of magnitude 9. It may not be capable of producing one, given the geometry of the subduction interface and the apparent weakness of the plate coupling. The operational seismology community is, in the current literature, divided on whether the Mariana Arc poses a megathrust threat at the magnitude 9 scale. The honest answer is that we do not know. The historical record is short, the instrumented record is shorter, and the geometry of the subduction is unusual enough that direct analogies to Cascadia or to Sunda are not reliable. But the historical record of Storegga is not about the specific subduction zone. The historical record of Storegga is about what happens to a coastal population that has not built its risk model around a low-probability, high-consequence event. The Mesolithic inhabitants of Doggerland did not have a tsunami early warning system. They did not have evacuation routes. They did not have a national-level emergency management agency that had run drills for the event. They did not have any of the institutional infrastructure that modern coastal populations have. And they were killed at scale. The modern Pacific Basin does have all of those institutional infrastructures. The National Tsunami Warning Center in Palmer, Alaska, and the Pacific Tsunami Warning Center in Ewa Beach, Hawaii, are both operated by NOAA and provide near real-time warnings to Pacific Basin coastal populations. The Federal Emergency Management Agency has, in collaboration with the United States Geological Survey, the National Tsunami Hazard Mitigation Program, and several state-level agencies, conducted detailed tsunami risk assessments for the United States Pacific Coast, Alaska, Hawaii, American Samoa, Guam, and the Commonwealth of the Northern Mariana Islands. The assessments are recent, and they are explicit. We are going to walk through what they say in Part 5. But here is the calibration point I want you to take into Part 5. even with all of the modern institutional infrastructure. The modern Pacific Basin risk assessments published by FEMA and the partner agencies in September of 2025 estimate that 79% of the projected economic losses from a major Pacific tsunami event, integrated across all the United States Pacific coast and territorial communities, would be associated with loss of life. 79%. Not destroyed property. Not displaced populations. Loss of life. The dollar figure attached to the loss of life component of the projected losses is $790 million per year on a long-term annualized basis. That number is the modern equivalent of the Storegga calibration. The modern Pacific risk model assumes that, despite the warning systems, despite the evacuation routes, despite the institutional infrastructure, a major tsunami event would still cause large-scale loss of life. The modern Pacific risk model has not solved the Storegga problem. The modern Pacific risk model has reduced the casualty count from what it would have been without the warning systems, but it has not reduced it to zero. The risk of mass casualty from a Pacific Basin tsunami event is, in the operational FEMA literature, treated as a serious enough threat to be modeled, costed, and prepared for. As of the most recent FEMA risk assessment document, the long-term annualized expected loss to United States Pacific coast and territorial communities is $1 billion per year, of which 79% is loss of life. The Mariana Trench is one of the nodes in the basin that the FEMA risk model is built around. The Hadal microbiology sits on top of one of those nodes. The 89.4% novel species inventory the MIR team published is a snapshot of the chemical state of the input to one of those nodes. The trench is alive in every sense that matters. Biologically alive, geologically alive, seismically alive. And the question of what the trench will do next, on the human timescale, is not a question that the science can answer with certainty. What the science can do is tell us where the risk lives, and the risk, on the operational basis of the FEMA risk assessments and the published Pacific tsunami forecasting literature, lives in the Pacific Basin's coastal populations. Part 5 is the part where we walk through who is in the path. By population, by state, by projected casualty exposure, and by what the institutional infrastructure says about the warning windows they would actually have. Part 5, the United States Coast Inventory. Let me start with the population number. According to the United States Census Bureau, in a 2019 analysis of population by proximity to the coastline, 94.7 million Americans live in counties that the Bureau classifies as coastline regions. That is the most conservative count. A broader definition, used by the National Oceanic and Atmospheric Administration, includes all coastal counties, which expands the number to 127 million Americans as of the most recent count, rising to 131 million as of 2022. By the broader definition, 31% of the United States population lives in a coastal county. By either definition, the number is in the range that this video's title described as 90 million coastal Americans, and the broader number is now nearer to 130 million. Of those 130 million coastal Americans, the subset that is exposed to Pacific Basin tsunami risk specifically includes the populations of the United States Pacific Coast states, the Pacific Territories, and the Caribbean Territories, whose tsunami risk profile is connected to Pacific Basin sources through the geography of plate motion and submarine landslide propagation. The states involved are California, Oregon, Washington, Alaska, and Hawaii. The territories involved are American Samoa, Guam, the Commonwealth of the Northern Mariana Islands, Puerto Rico, and the United States Virgin Islands. The combined population of all those states and territories runs into the tens of millions, with California alone accounting for approximately 26.8 million coastal county residents as of the most recent Census Bureau release. Now, here is where the FEMA risk assessment becomes operationally specific. In September of 2025, the Federal Emergency Management Agency, in collaboration with the United States Geological Survey, the National Tsunami Hazard Mitigation Program, and several partner agencies, published a document titled Estimated Average Annualized Tsunami Losses for the United States. The document, identified by the catalog number Jesus P2426, is a publicly available risk assessment study that estimates the expected losses to United States coastal communities from tsunami events, averaged across all probable scenarios and integrated over a long-term horizon. The document's headline finding is that the long-term annualized expected tsunami loss to United States coastal communities is approximately $1 billion per year. The disaggregation of that $1 billion is what the rest of this part is about. Of the $1 billion per year, the document attributes $790 million per year, or 79% of the total, to losses associated with loss of life. The remaining 21% is associated with destroyed buildings, damaged infrastructure, displaced populations, and economic disruption. Read that distribution one more time, because it is the key sentence of the entire FEMA risk assessment exercise. 79% of the projected tsunami losses in the United States, integrated across all coastal communities and all probable scenarios, are losses of human life. Pause for a second on that sentence because it is the sentence this video has been building toward. The Federal Emergency Management Agency, the United States Geological Survey, the National Tsunami Hazard Mitigation Program, and the partner agencies have, in their published, peer-reviewed, formally adopted risk assessment document, attributed 79% of the projected national tsunami losses to loss of life. The risk to United States coastal communities from tsunamis is, in the formal operational assessment of the United States federal government's emergency management apparatus, primarily a risk to human lives. The destroyed property component is, in dollar terms, a fifth of the total. The lives component is 4/5. The geography of where those projected losses concentrate is also documented in the FEMA risk assessment document. The state of Oregon, in the analysis, is identified as having a specific risk profile that produces some of the highest projected fatality-related losses in the entire study. The reason, as the document explains, is the combination of geographic exposure and warning time geometry. The Oregon coastline is exposed to the Cascadia subduction zone, where the rupture of a major megathrust event would generate a locally sourced tsunami that arrives at the Oregon shoreline within 15 to 20 minutes. The warning window for the Oregon coastal population, in the event of a Cascadia rupture, is shorter than the typical evacuation time required to move a coastal population from the inundation zone to the safe elevation zone. The result, in the FEMA risk model, is high projected fatality exposure for the Oregon coast. In the operational language of the FEMA document, in 41 separate scenarios that the model evaluated, at least one resident of a United States coastal community would have insufficient time to evacuate before wave arrival. 41 scenarios. Each scenario is a specific combination of source, wave height, and target community. The 41 scenarios are not all the worst-case scenarios. They are the scenarios that, on the median assumptions of the model, would still produce some unavoidable fatality exposure. The worst-case scenarios, which the document analyzes separately, produce fatality counts in the thousands. The Pacific territories carry their own specific risk profile. Guam and the Commonwealth of the Northern Mariana Islands, both United States territories, sit on the western, overriding plate side of the Mariana Trench. The distance from the trench axis to the closest inhabited shorelines is, as I described in Part 3, less than 200 kilometers. The locally generated tsunami arrival time, in the event of a moderate magnitude earthquake along the Mariana subduction interface, is between 10 and 45 minutes. That is shorter than the Oregon warning window. The Guam and Commonwealth of the Northern Mariana Islands populations would, in the event of a locally generated Mariana tsunami, have less time to evacuate than the Oregon coastal population would have in the event of a Cascadia rupture. This is the part of the Pacific Island Times article that I referenced in Part 3, made operationally specific. The scientists cited in that article: Landon Aidlet at the National Weather Service Guam office, Laura Kong at the International Tsunami Information Center, and Marie Abel, a physical oceanographer who has spent decades studying Pacific Basin wave propagation, all stated, on the record, that the Mariana Trench is a local source for tsunamis. The trench does not dampen wave energy. The trench does not protect the islands above it. The trench, in fact, concentrates the seismic activity of the subduction interface directly beneath the inhabited islands, and any wave generated by that subduction activity arrives at the island's shorelines within minutes. The article also documented a historical analogy that is worth holding in mind. In September of 2009, a magnitude 8.1 earthquake occurred along the Tonga Arc subduction zone in the South Pacific. The earthquake generated a tsunami that, when it reached the coastlines of Samoa, American Samoa, and Tonga, produced run-up heights of more than 20 meters in some locations. The fatality count was approximately 200 people across the affected coastlines. The 2009 Samoa event, in the operational analysis, disproved the local belief that the Tonga Trench, like the Mariana Trench in the Western Pacific, somehow protects the islands above it from tsunamis. The Tonga Trench in the 2009 event was the source. The wave traveled the short distance to the islands. The population had minutes of warning at best. 200 fatalities in the 2009 Samoa event is not a number on the scale of Storegga or of the 2004 Indian Ocean event. It is, however, 200 specific individuals whose deaths can be traced to a single submarine seismic mechanism, a single short warning window, and a single local population that had not built its risk model around the possibility. The 2009 Samoa event is the operationally most recent example of what a local tsunami in the Western Pacific actually does, and the operational lesson, as the National Weather Service Guam office, the International Tsunami Information Center, and the operational oceanography community have been saying publicly since at least 2010, is that the Mariana Trench is a comparable risk. I want to be precise about what comparable means. Comparable does not mean identical. The Mariana subduction zone is not the Tonga subduction zone. The geometry is different. The plate interface coupling is different. The historical record of large events is different. Comparable means that the operational mechanism is the same. A subduction interface earthquake of moderate or greater magnitude, occurring less than 200 kilometers from inhabited shoreline, generates a tsunami that arrives at the shoreline in a warning window measured in minutes, not hours. The Tonga arc proved the mechanism. The Mariana arc, in the assessment of the National Weather Service Guam office, and the International Tsunami Information Center, has the same mechanism available. The 41 scenarios in the FEMA risk model that produce unavoidable fatality exposure include in their structure scenarios that involve locally generated tsunamis in the Pacific territories. The total projected fatality exposure, integrated across all 41 scenarios, is the basis for the $790 million annual loss of life component of the larger $1 billion annual loss estimate. The Mariana subduction zone is one of the sources that contributes to that aggregate. The Tonga arc, the Aleutian arc, the Kuril arc, the Cascadia subduction zone, the Mexican subduction zone, the Peruvian and Chilean subduction zones, the Sunda arc, and several smaller systems all contribute as well. Now, I want to step back and walk through the operational warning system. Because the warning system is the part of the institutional infrastructure that most directly shapes the projected fatality count. The National Tsunami Warning Center in Palmer, Alaska, monitors seismic activity along the entire Pacific Rim, plus the Aleutian and Alaskan source regions, and is responsible for issuing tsunami warnings to the United States Pacific Coast states and the Alaskan coast. The Pacific Tsunami Warning Center in Ewa Beach, Hawaii, monitors seismic activity across the broader Pacific Basin and is responsible for issuing tsunami warnings to Hawaii, the United States Pacific Territories, and through international coordination agreements to most Pacific Rim nations. Both centers are operated by NOAA. Both centers operate 24 hours a day, 7 days a week. Both centers have access to the global seismic network, which provides earthquake detection and magnitude estimation in near real time, typically within 2 to 5 minutes of the originating event. The warning system works, in the operational sense, by detecting the earthquake first, estimating the magnitude and location, modeling the expected tsunami propagation across the basin, and issuing warnings to the predicted affected coastlines. The system has been continuously improved since the 2004 Indian Ocean event, which exposed gaps in the warning coverage of the Indian Ocean Basin. The Indian Ocean Basin now has its own warning system, integrated with the Pacific System. The Caribbean Basin has its own. The Atlantic Basin has its own. The Mediterranean Basin has its own. The global tsunami warning infrastructure, as of 2025, is the most developed it has ever been. But the warning system, as the operational community will tell you if you ask carefully, has a fundamental geometric limit. For a tsunami sourced locally within a few hundred kilometers of the affected shoreline, the warning system cannot deliver a useful warning. The earthquake propagates as a P wave at approximately 6 kilometers per second. The tsunami propagates across the deep ocean at approximately 200 meters per second, which is the speed of a passenger jet. The S-wave shaking reaches the affected shoreline before the warning system can issue a formal warning. The tsunami arrives minutes after the shaking ends. For a population within 50 to 200 kilometers of the source, the only useful warning is the shaking itself. If you feel the ground shake on a coastal area near a subduction zone, the operational guidance is to immediately move to high ground. Do not wait for an official warning. The official warning will not arrive in time. That guidance is the part the operational community emphasizes in every public training and every educational outreach effort. The shaking is the warning. The institutional warning system, valuable as it is designed for distant tsunamis. For local tsunamis, the population is dependent on its own situational awareness. If you live on a Pacific coastline within a few hundred kilometers of a subduction zone, and you feel a strong earthquake, your operational guidance is to immediately move to high ground without waiting for any further information. The populations exposed to this geometry in United States territory specifically include the Cascadia coastal communities of Oregon and Washington, the Southern Alaskan coastal communities, the Hawaiian coastal communities, the Puerto Rican and United States Virgin Islands coastal communities, the American Samoan coastal communities, the Guam coastal communities, and the Commonwealth of the Northern Mariana Islands coastal communities. The combined population of those communities is in the millions. The combined operational situational awareness capability of those communities is the variable that, in the FEMA risk assessment model, determines how many of the $790 million annual loss of life projection becomes an unavoidable number, and how many can be reduced by drills, by education, by infrastructure improvements, by zoning that moves residential populations out of the inundation zone. The United States National Tsunami Hazard Mitigation Program, since its founding in 1996, has been working on exactly that reduction. The program has funded tsunami inundation mapping for every United States Pacific coastal community. The program has supported the development of evacuation route signage, of community drills, of school curricula that teach children to recognize the shaking and to evacuate without waiting for instruction. The program has supported the deployment of the Deep Ocean Assessment and Reporting of Tsunamis system, the DART buoys, which provide real-time deep ocean tsunami detection in the Pacific Basin. The DART buoys, as of 2024, were 39 instruments deployed across the Pacific, the Atlantic, and the Caribbean. Each DART buoy is a pressure sensor on the deep sea floor, communicating with a surface buoy that relays the data via satellite to the warning centers. The infrastructure is real. The program is real. The training is real. The DART buoys are real. And the FEMA risk assessment, which incorporates all of those mitigations into its projection, still concludes that 79% of the annualized projected tsunami losses to United States coastal communities are losses of life. The infrastructure has not solved the problem. The infrastructure has reduced the problem to the level that the FEMA assessment now describes as residual unavoidable risk. The residual unavoidable risk is $790 million per year in projected loss of life value. Translated into expected lives lost per year on long-term average, the figure runs into the tens or hundreds, depending on the assumptions of the underlying actuarial model. Tens to hundreds of expected lives lost per year, on average, across United States Pacific coastal communities, after all the warning systems and all the institutional infrastructure that has been built. That is the modern operational baseline, and the Mariana subduction zone, the trench whose biology the MIR team systematically sampled in 2021 and published in March of 2025, is one of the sources that contributes to the aggregate. The audience for this channel has now walked through every part of the operational case. The biology in Part 1 and Part 2, the mechanism in Part 3, the historical parallel in Part 4, the modern risk profile in Part 5. What remains is the part of every honest analysis that the rest of the coverage usually skips. The counter-arguments, the genuinely uncertain parts, the places where the operational consensus is not as settled as the rest of the case might suggest. That is Part 6. Part 6, the honest caveats. I want to do something in this part that almost no piece of long-form coverage of these topics does. I want to walk you through, carefully and in detail, what the operational seismology, oceanography, and biology communities are not yet certain about. Because the case I have been building, across the previous 5 parts, depends on the strength of the underlying science, and the underlying science, while substantial, is not monolithic. There are places where the consensus is settled. There are places where the consensus is forming. And there are places where, frankly, the science does not yet support the inference that an honest review of the operational record would suggest. I want to be precise about which is which. Let me start with what is genuinely settled. The mere dataset is real. The 89.4% novelty figure is the published result of a peer-reviewed paper in the journal Cell. The methodology of the mere sequencing pipeline has been independently scrutinized by the editorial process of Cell, which is one of the most rigorous peer review processes in the biological sciences. The genetic sequences are publicly deposited and can be queried by any laboratory that has the bioinformatics capability. The headline finding that the Hadal microbial community is largely composed of organisms not previously sequenced is not in dispute in the operational biology community. The implication that the chemistry of the Hadal zone is being mediated by an ecological assembly that has not previously been inventoried is also broadly accepted, though the specific magnitudes of the biogeochemical effects are still being quantified. What is less settled on the biology side is the question of how much the mere discovery actually changes the operational models of global ocean biogeochemistry. The Mir team's argument in the supplementary materials of the 3rd paper is that the Hadal community is mediating chemistry that affects subduction zone outgassing. The argument is plausible. The quantitative support for the argument, however, depends on assumptions about how much Hadal sediment is actually subducted versus remaining on the overriding plate. Those assumptions are reasonable, but not yet independently verified by direct measurement. A genuinely careful reading of the Mir papers would conclude that the biology is real, the chemistry is novel. But the planetary scale geochemical implications are an active area of research, not a settled conclusion. On the seismology side, the case is similar. The Tectonophysics 2025 paper on intermediate depth seismicity in the southernmost Mariana subduction zone is published, peer-reviewed, and part of the formal scientific record. The pattern of intermediate depth earthquakes correlating with hydration of the subducting plate is supported by the paper's analysis. The interpretation of that pattern as direct evidence of water transport into the mantle is consistent with the broader subduction zone literature, including independent studies of the Cascadia and Japan subduction zones that have reached similar conclusions about the role of water in slab metamorphism. What is less settled on the seismology side is the question of how the intermediate depth seismicity pattern relates to the shallow seismicity that would generate a damaging tsunami. The shallow part of the subduction interface, where a megathrust earthquake would occur, is mechanically distinct from the intermediate depth zone where the dehydration earthquakes are happening. The two zones are connected, in that they are part of the same subducting slab. But the timing relationships between activity in one zone and activity in the other are not well established. A genuinely careful reading of the seismology literature would conclude that the Mariana subduction zone is active, that the hydration of the subducting plate is documented, and that local tsunamis are possible, but that the specific probability of a large magnitude shallow interface event in the near term is not predictable on the basis of current science. The Mariana megathrust risk, specifically, is the part of the case where the science is most genuinely uncertain. The historical record of large earthquakes in the Mariana Arc, as I noted in Part 3, is thin. The instrumented record has not captured a magnitude 8 or larger event. The geometry of the subduction interface, with its near vertical slab descent and its apparent weak coupling, suggests that the arc may be capable of generating only moderate magnitude earthquakes, not the megathrust scale events that would produce a basin-crossing tsunami. The operational seismic hazard models for the Western Pacific, which I have reviewed in the published literature, generally treat the Mariana Arc as a lower magnitude source than the Cascadia or Sunda arcs. So, when I tell you, in this video, that the Mariana Trench is a local source for tsunamis, that statement is correct, but it should be heard with the precision the operational science supports. The Mariana Trench is a local source for tsunamis on the order of the 2009 Samoa event, which killed approximately 200 people. The Mariana Trench is not, on the current evidence, a likely source for a basin-crossing megathrust event on the order of the 2004 Indian Ocean tsunami, which killed approximately 230,000 people. The two scales of risk are very different, and the honest scientific case for paying attention to the Mariana subduction zone, specifically, rests on the local tsunami component, not on the basin-crossing component. That said, the Pacific Basin as a whole does contain multiple subduction zones that are, on the current science, more likely sources for a basin-crossing megathrust event than the Mariana Arc. The Cascadia subduction zone is the most studied and best understood of those. The probabilistic seismic hazard assessment for Cascadia, as published in the most recent versions of the United States Geological Survey's National Seismic Hazard Maps, includes a substantial probability of a magnitude 8 or larger event in the coming decades, with corresponding tsunami implications for the United States Pacific Northwest. The Sunda Arc, which produced the 2004 event, has not, on the current science, exhausted its capacity to produce additional large events. The Tohoku region of Japan, which produced the 2011 event, also has not. The aggregate probability of a basin-crossing Pacific tsunami event, integrated across all the major subduction sources on a multi-decadal horizon, is substantial. The honest framing, then, is that the Mariana Trench is one of multiple Pacific subduction sources. The operational seismology community has never seen, in the instrumented record, a Mariana megathrust event of the scale that would justify a basin-crossing tsunami warning. That is not a guarantee, because the instrumented record is short. But it is the strongest claim the data currently supports. The Mariana Trench's biology is now, for the first time, systematically documented. The Mariana Trench's seismicity is active, hydration-driven, and capable of generating local tsunamis. The Mariana Trench is not, on the current science, the most likely source for the next major Pacific tsunami. But the Mariana Trench is part of the same basin, the same plate tectonic system, and the same operational risk framework as the more studied sources. The case for paying attention to the Mariana Trench specifically is not that it is uniquely dangerous. The case is that the Pacific Basin as a whole is connected. The operational risk model for the basin is well developed, and the recent advances in our understanding of the Mariana Trench's biology, mechanics, and chemistry are all advances that improve our understanding of the basin as a whole. There is one more set of caveats I want to address before closing this part. The connection I made between the mere biological discovery and the seismic risk profile is the kind of cross-disciplinary inference that requires care. The biology paper and the seismology paper, as I noted earlier, were published in different journals and address different questions. The connection between them, that the Hadal microbes are processing the chemistry of the input to the subduction conveyor, is a connection that the mere supplementary materials make explicitly. The further connection, that the chemistry being processed is also chemistry that influences the seismic behavior of the descending plate, is a connection that the operational geochemistry community has been building for at least a decade, but which has not yet, to my knowledge, been quantified in a single peer-reviewed publication that brings all the elements together. The connection is, in other words, plausible, supported by the individual pieces of evidence, but not yet formalized as a single coherent published model. The honest framing of the connection in this video is that the biology and the seismology are both happening at the same location. Both involve water and the chemistry of the subducting plate, and both are matters of active research. The further inference that the biology and the seismology are mechanistically coupled in a way that affects the timing or magnitude of seismic events is a hypothesis that the operational research community is investigating but has not, as of May 2026, established with the kind of certainty that would justify treating it as a settled fact. I want to be especially careful about this because the cross-disciplinary inference is exactly the kind of inference where, in mainstream coverage, claims often get overstated. A video that promised, in its title, that the Mariana Trench had just done something nobody alive had ever seen, and then connected that to a tsunami threat, could very easily slip into the kind of overstatement that the operational science would not support. I have tried, in this video, to keep the inferences within what the published literature supports. The mere discovery is real and unprecedented in its biological inventory scale. The Mariana subduction zone is active and capable of generating local tsunamis. The Pacific Basin tsunami risk is documented and quantified by FEMA. These are all true statements, individually, supported by the published record. The connections between them, which this video has drawn out, are connections that the operational research community is building, but they are not yet fully integrated into a single formal model. What this means, in practical terms, is that the audience for this channel should treat the video's narrative as a synthesis of multiple, individually well-supported lines of evidence, with the integrated picture being one that the science is approaching but has not yet fully completed. The synthesis is honest. The synthesis is responsible. The synthesis is, I believe, the right way to think about what is happening in the Pacific Basin in 2026. But the synthesis is not, on the current state of the published record, a settled scientific conclusion. It is an emerging consensus, and emerging consensus, while valuable, is not the same as settled fact. The audience for this channel has now spent 90 minutes thinking carefully about the Mariana Trench, the Hadal microbial community, the Pacific subduction zone risk profile, and the historical record of submarine geological events. That is more time than almost any audience anywhere has spent on this material in a single sitting. The conclusions you have arrived at, having sat with the material this long, are conclusions that you should hold with a calibrated sense of uncertainty. The science is real. The science is incomplete, and the operational community will continue to refine the picture in the years ahead. That brings us to Part 7. Part 7, the next 12 months. I want to spend the next section walking you through, concretely, what the operational research community is going to be doing over the next year, and what the audience for this channel should be watching for. Because the mere discovery is not the end of a research program. It is the beginning. And the next 12 to 18 months on the published research calendar are going to produce a sequence of follow-on findings that, when they arrive, will either reinforce the picture this video has built or modify it. I want you to know, in advance, what those findings will be, where they will be published, and what to look for in the language of the papers when they come out. The first item to watch is the next sequencing batch from the MIR consortium. The March 2025 cell papers were based on the August through November 2021 Fendouzhe expedition. The consortium has not stopped sequencing. The published roadmap in the supplementary materials of the third cell paper indicates that the team is working through additional sediment samples from the same expedition that were held in reserve, and that follow-on expeditions are planned through the Chinese Deep-Sea Research Program. The next major MIR publication, when it arrives, is likely to focus on what the consortium calls the deep sediment fraction, which is the sediment below the top 10 centimeters of the trench floor. The deep sediment fraction is the part of the column that, on geological timescales, is closest to being subducted. The microbiology of that fraction is, by the consortium's own framing, the most direct chemical input to the subduction conveyor. When that paper appears, you should expect to see the 89.4% novelty figure either reaffirmed or revised, and you should expect new metabolic pathway findings that connect the deep sediment community to the chemistry of the descending plate. The publication venue is likely to be Cell Again, or one of its sister journals in the Cell Press family. The expected publication window, based on standard manuscript preparation timelines from the existing supplementary materials, is the second half of 2026 into the first half of 2027. The second item to watch is the follow-on work by Caladan Oceanic. Victor Vescovo, founder of Caladan Oceanic, completed his 15th descent to Challenger Deep in the spring of 2021 and has not stopped expanding his Triton submersible's operational record. In October of 2024, Vescovo led the first-ever human descents into the Yap Trench and the Palau Trench, two trenches in the western Pacific that, prior to those dives, had never been visited by a human. The Yap and Palau dives are the operational extension of the 5 Deeps expedition. They are also, in the longer-term context of the Caladan Oceanic mission, the first systematic effort to inventory the deep biology and geology of the smaller western Pacific trenches that, until 2024, had been entirely unsampled. The audience for this channel should watch, over the next 12 months, for the first peer-reviewed publications based on the Yap and Palau samples. The Caladan Oceanic operations are typically accompanied by scientific teams from the partner institutions, including the Woods Hole Oceanographic Institution and the Schmidt Ocean Institute. Papers based on the 2024 dives are likely to appear in deep-sea research, in marine geology, and in the broader subduction zone literature. When they appear, you should expect to see the MIR methodology, the metagenomic sequencing of trench sediments, applied to the smaller western Pacific trenches. Whether the 89.4% novelty rate replicates in those smaller trenches will be the test of whether the Mariana finding is unique to that specific trench, or whether it is, as I suspect, a feature of Hadal zone biology generally. The third item to watch is the Schmidt Ocean Institute's 2025 and 2026 expedition calendar. Schmidt Ocean Institute, operating the research vessel Falkor, has been conducting deep-sea expeditions across the global ocean basin for over a decade. Their 2025 calendar includes specific expansions of Mariana Trench coverage, with the remotely operated vehicle Subastion conducting dives that complement the manned submersible work of the Caladan Oceanic and Chinese programs. The Falkor expeditions are publicly streamed on the Schmidt Ocean Institute's website and through scientific outreach channels. The data from those expeditions, when published, will appear in the standard deep-sea oceanographic journals. The audience for this channel should bookmark the Schmidt Ocean Institute's news feed. Over the next 12 months, that feed will carry, in close to real time, the announcement of any major findings from their 2025 and 2026 Mariana expeditions. The fourth item to watch is the FEMA Risk Assessment Update Cycle. The HAZUS P2426 document I cited in Part 5 was published in September of 2025. The FEMA Risk Assessment Cycle for major hazard categories typically produces updated documents on a 2 to 3 year cadence. The next major update to the United States Tsunami Risk Assessment is expected in late 2027 or early 2028. Between now and then, FEMA and the partner agencies will publish smaller supplementary studies, scenario-specific analyses, and updates to the inundation mapping for individual coastal communities. The audience for this channel should watch the FEMA HAZUS catalog page and the National Tsunami Hazard Mitigation Program publications page. Smaller technical updates to the risk model will appear there before any major revisions reach the headline-level HAZUS document. The fifth item to watch is the Cascadia Subduction Zone, Probabilistic Seismic Hazard Update. The United States Geological Survey publishes the National Seismic Hazard Maps on a 6-year cycle. The most recent revision was the 2023 edition, which incorporated updated probabilistic estimates for the Cascadia Subduction Zone. The next major revision is scheduled for 2029. Between now and then, United States Geological Survey will publish smaller updates as new paleoseismic data is integrated into the model. The audience for this channel should watch for any new paleoseismic papers from the Pacific Northwest, particularly papers based on coastal sediment cores and tsunami deposits in Oregon and Washington. Those papers, individually, can shift the probabilistic estimates by meaningful amounts. When they appear, you should expect them in journals like Geology, Quaternary Science Reviews, and Earth and Planetary Science Letters. The sixth item to watch is what the operational community calls the Lessons Learned Literature on the July 2025 Kamchatka event. The magnitude 8.8 earthquake off the Kamchatka Peninsula and the basin-crossing tsunami it generated was the largest Pacific tsunami source event since the 2011 Tohoku earthquake. The operational community is currently in the process of analyzing the warning system performance, the modeled versus observed wave heights, and the implications for the global tsunami forecasting infrastructure. The lessons learned papers from that event will begin appearing in peer-reviewed venues over the next 12 to 24 months. They will modify, in incremental ways, the forecasting parameters that the National Tsunami Warning Center and the Pacific Tsunami Warning Center use to issue warnings. The audience for this channel should expect those papers in the Journal of Geophysical Research, in Pure and Applied Geophysics, and in the technical proceedings of the International Tsunami Information Center. The seventh item to watch is something almost nobody outside of the operational geochemistry community is talking about yet: the cross-disciplinary integration of the mere biological data with the tectonophysics seismic data, with the operational oceanographic data, into a single coherent model of the trench as a coupled biogeochemical geophysical system, is the kind of integration that will, when it happens, change the textbooks. As of May 2026, that integration has not been published. The pieces are all in the literature. The synthesis is not. But there are research groups at Shanghai Jiao Tong University, at Woods Hole, at Scripps Institution of Oceanography, at the Lamont Doherty Earth Observatory, that are working on the synthesis. When the first paper that brings the biology, the seismology, and the geochemistry into one model appears, it will be the paper that defines the next decade of Hadal Zone research. The audience for this channel should watch the high-impact journals Nature, Science, Cell, Proceedings of the National Academy of Sciences for that paper. It is coming. The publication window is, on the typical pace of cross-disciplinary work of this complexity, in the 18 to 36-month range. When it arrives, the audience for this channel will recognize it. Almost nobody else will. Think about what it means to be calibrated to recognize a paper like that in advance. It means that when the announcement comes, you will not need a journalist to explain it to you. You will know what the paper is, why it matters, what came before it, and what it implies. You will be able to read the abstract and immediately understand which existing models it modifies. That is the kind of pattern recognition this channel exists to build. Not in the abstract. In the concrete operational sense of being able to anticipate which announcements, in the next 12 to 18 months, will be the announcements that matter. The eighth and final item to watch is something the operational community is more reluctant to discuss publicly, but which the literature supports. The Pacific Basin has not produced its maximum credible Pacific Basin tsunami event in living memory. Each of the major subduction sources, Cascadia, Sunda, Tohoku, Aleutian, Kuril, Chilean, Peruvian, has a probabilistic hazard envelope that includes, on a multi-decadal horizon, the possibility of a major event. The combined probability across all sources, integrated over the next 20 years, is substantial. The operational community does not predict, on the basis of current science, which source will rupture first or when. But the community does treat the basin-wide probability as serious enough to justify the existing warning infrastructure, the FEMA risk assessment documents, the DART buoy network, the inundation mapping, and the public education outreach. The audience for this channel should hold that combined probability in mind, not as a source of dread, but as an operational backdrop against which the next 12 months of research findings will be evaluated. The next 12 months are going to produce a steady stream of findings. The mere follow-on. The Caledon papers from Yap and Palau. The Schmidt Ocean Institute expedition reports. The Kamchatka lessons learned literature. The FEMA supplementary documents. The Cascadia paleoseismic updates, and at some point, possibly within 12 months, possibly longer, the first integrated biogeochemical geophysical model of a Hadal Zone subduction trench. Each of those findings will be a small adjustment to the picture this video has built. None of them, individually, will redefine the picture, but cumulatively, over the next 2 to 3 years, they will produce a new operational understanding of the trench that is meaningfully different from what we have now. That is what the next 12 months looks like. That is what to watch for. That is the operational forecast that this channel exists to provide. And that is the bridge into the close. In March of 2025, a research team based at Shanghai Jiao Tong University, working with collaborators at the University of Copenhagen and Lawrence Berkeley National Laboratory, published in the journal Cell, the largest single addition to the catalogued biological inventory of the deep ocean in the history of marine biology. 7,564 microbial species. 89.4% of them previously unknown to science. The discovery was made in the sediments of the Mariana Trench, between 6,000 and 10,900 meters below the surface of the western Pacific Ocean. During a Chinese expedition that completed 33 submersible dives across August through November of 2021. The headline of that discovery is the headline this video began with. The discovery was substantial in its biological scale. It is, the journal Cell editorial concluded, comparable in scientific weight to the Human Microbiome Project. It will, over the coming 3 to 5 years, lead to revisions of the global biogeochemistry models that describe how the deep ocean processes the chemistry of the surface ocean and feeds it into the planet's geological cycles. It is, by any honest measure, the most consequential single advance in deep-sea biology of the modern era. The same trench, on the same Pacific tectonic plate, is also one of the most seismically active subduction zones in the world, with a documented history of intermediate-depth earthquakes that map the transport of water from the surface ocean into the mantle. It is a local source for tsunamis in the operational assessment of the National Weather Service Guam office, the International Tsunami Information Center, and the broader operational oceanography community. The warning window for a locally generated Mariana tsunami, when one arrives, is between 10 and 45 minutes. The inhabited shorelines closest to the trench, on Guam and the Commonwealth of the Northern Mariana Islands, are United States territories. The United States coastal population at risk from Pacific Basin tsunami events, integrated across all sources, is approximately 94.7 million in the conservative Census Bureau definition, and approximately 130 million in the broader NOAA definition. The Federal Emergency Management Agency, in its September 2025 risk assessment document, attributes 79% of the projected annualized United States tsunami losses to loss of life. $790 million per year on long-term average in projected loss-of-life value. The institutional infrastructure for tsunami warning is the most developed it has ever been. The infrastructure has reduced the risk, but has not eliminated it. The residual unavoidable risk is the $790 million component of the $1 billion annual aggregate. The Storegga slide, 8,200 years ago, killed up to 25% of the Mesolithic population of northern Britain in a single morning. It eliminated the inhabited land of Doggerland. It is the only event in the Holocene archaeological record at the combined scale of magnitude and direct coastal impact that the Mariana Trench, in the right combination of seismic and submarine landslide circumstances, could in principle approach. The Mariana Trench is not, on the current science, likely to produce a Storegga-scale event in the near term. But the Pacific Basin as a whole contains multiple subduction sources that are capable of producing basin-crossing megathrust events. The Cascadia, the Sunda, the Tohoku, the Aleutian, the Kuril, the Chilean. Each is studied. Each is monitored. None has, in the instrumented record, produced its maximum credible event in living memory. The honest synthesis is this. The Mariana Trench has, in the last 4 years, been transformed in our scientific understanding from a place of curiosity to a place of intense systematic investigation. The biology of the trench is now inventoried at unprecedented scale. The geology of the trench is better mapped than it has ever been. The seismicity of the trench is being analyzed with the most current intermediate depth earthquake methods. The chemistry of the trench is being connected, in the published literature, to the planetary geochemistry cycle, and the operational tsunami risk profile of the Pacific Basin, of which the Mariana Trench is one node, is being assessed by the United States federal government with the most current actuarial and hazard modeling tools available. The trench has not done one single thing that nobody alive has ever seen. The trench has done thousands of things, in the form of the 89.4% novel species inventory, in the form of the intermediate depth earthquake patterns now being mapped, in the form of the chemical pre-processing that the MIR team has begun to quantify. Each of those thousands of things, individually, is something the operational science community had never seen at the level of detail and systematic coverage that the MIR Consortium and the Tectonophysics 2025 team have now provided. The trench is, in 2026, more active in the scientific record than it has been at any prior time in human history. Not because the trench itself is changing, but because we are finally, after a century of deep-sea exploration, paying it the systematic attention it warrants. The audience for this channel has just spent 2 hours thinking carefully about what the Mariana Trench actually is, what the research community is actually finding, what the historical record actually shows, and what the modern risk profile actually looks like. That is a calibrated investment. The audience for this channel is, after this video, calibrated to recognize the next major announcement out of the Mariana Trench research community. The next major paper on Pacific Basin tsunami risk. The next major intermediate depth seismicity finding. The next major Hadal zone biological discovery when they appear. The pattern recognition that this video has tried to teach is the pattern recognition that will, in the years ahead, let you read those announcements with the context that almost nobody else watching the broadcast will have. That is what this channel exists to do. That is why we spend 2 hours on a single topic instead of 2 minutes. That is why we cite the journal name and the publication date and the lead author's affiliation. That is why we walk through the caveats in Part 6 instead of leaving them in the footnotes. The audience for this channel has, by virtue of paying attention, just become one of the small minority of people anywhere in the world who can read the MIR papers. The Tectonophysics 2025 paper, the FEMA risk assessment document, and the operational tsunami forecasting literature, and place them into a single coherent picture. That picture is the picture of a Pacific basin whose risks and whose wonders are now, more than ever, intertwined, and whose future, on the operational human timescale, will be more visible to this audience than to almost anyone else. The next event in the Pacific Basin will come. The next paper from the MIR Consortium will come. The next intermediate depth seismicity finding will come. The next tsunami advisory, for whatever source and whatever shoreline, will come. We are going to be watching this all summer and into the years ahead. We are going to be tracking what the trench does next. We are going to be paying attention to the parts of the science that most of the news cycle will not have time to walk through carefully. If you have watched all the way to here, you have done something almost nobody alive has ever done with material this technical. You have sat with 7,564 microbial species, 3,500 cubic kilometers of submarine landslide, 94.7 million coastal Americans, $790 million of annualized projected loss of life, 10 to 45 minutes of local Mariana tsunami warning, and 89.4% of a biological inventory that the modern science is still in the process of integrating. You have sat with all of it. You have followed the inferences. You have, I hope, been able to evaluate them on your own terms. That is the gift of paying attention. That is what this channel is here for. The trench is real. The biology is real. The seismicity is real. The risk is real. and the audience that has watched this video end to end is calibrated. The next thing the trench does, when it does it, will be visible to you in a way that it will not be visible to almost anyone else. And what it will be, when it arrives, is not for me to predict from May of 2026. The science is what it is. The history is what it is. The risk is what it is. The trench will continue to be studied. The Hadal microbial community will continue to be inventoried. The intermediate depth seismicity will continue to be mapped. The tsunami warning system will continue to be improved. And somewhere, on the Pacific Basin, on a Tuesday afternoon that will look like any other Tuesday afternoon when it begins, a coastal population will feel a sudden shaking. A few seconds, maybe more. The ground will lift, then settle. The people who feel it, the ones who have been paying attention, will know what to do. The people who have not been paying attention will hesitate. The tsunami warning system will broadcast an advisory. The DART buoys will record the wave. The Pacific Tsunami Warning Center will issue the alert. And the audience for this channel, wherever they are on the Pacific Basin at that moment, will already be moving to high ground. That is the question that does not go away when this video does. The question is not whether the next event will arrive. The question is whether the people who would otherwise be hesitating on the coastline closest to its source are now the people who have, by virtue of spending the last 2 hours with this material, become the people who do not hesitate. That is the only variable, in the end, that any of this analysis was ever about. And it is the variable that lives now with you.